Okay. Thank you so much, Justin and Martin, for organizing this and inviting me. Yeah, I'm really excited to talk about my work. I'll try to go as quick as I can for time, but feel free to interrupt me with questions during or at the end. Yeah, so I'm a new assistant professor at the University of Utah at the School of Medicine in the Department of Human Genetics. I've been here for about a year, and I've been adding a new technology to my lab, a new tool that essentially I've been the one driving. I have some help in my lab, which is to use AlphaFold to do protein-protein interaction screens. And that has developed into a big need for using the resources here at the University of Utah at the Center for High Performance Computing, running AlphaFold on lots of GPUs. So I'm going to talk about what I'm doing, why it's important, and my experience running these AlphaFold screens. So yeah, so the outline of my brief talk is just to talk generally about how we predict protein-protein interactions with AlphaFold and what that means, why it's important. And then I'll tell you about our experience running AlphaFold at the CHPC here at the University of Utah. So I'm just going to start very general and say that, our cells of all organisms are chock full of proteins. And those proteins mostly carry out their functions by interacting with other proteins, that they bind to other ones. So that's really important. And I have just a few examples of some extremely important protein-protein interactions to help illustrate this to everyone to get on the same page. The spike protein from SARS-CoV-2 binds to our ACE receptor, and that's how it enters cells and infects people. Insulin, a hormone in our blood binds to the insulin receptor, and that is critical for how glucose is taken up by cells. And then our muscles contract due to the binding between myosin and actin. So hopefully I've illustrated in a basic way that protein-protein interactions are essential for biology and how biology works. Last year, there was a Nobel Prize for protein structure prediction. The efforts of DeepMind and Google for developing AlphaFold. AlphaFold is an AI software that predicts the structure, the three-dimensional structure of a protein. You can give AlphaFold the primary amino acid sequence. So it's just a bunch of letters representing each amino acid, threonine, glycine, arginine, so forth. And it'll give you back the predicted 3D structure. And what's really important for my work, and which I'll talk about, is that you can get the three-dimensional structure of more than one protein interacting with one another. It doesn't just have to be three-dimensional structure of one protein. So I did a prediction of insulin, a hormone in our blood, interacting with our receptor. And you can see insulin in blue, just fitting in like a key in a lock. So that's this type of structure is very important for my work, seeing the interaction between proteins. And so what this means is that now we have a new tool, our computers, to predict protein interactions on a massive scale, to do something called a screen. We can take, say, our favorite protein, this blue protein, and we can systematically test whether it interacts with other proteins. And finding that magic key and lock relationship between our favorite protein and another protein is really important for us to help study what our favorite protein does, or to develop a new drug. So these types of screens with AlphaFold enable us to study biology in a new way. There are many experimental approaches that are similar to this, to do protein-protein interaction screens using biochemical techniques, but this is a new tool in our toolbox that we can use. It doesn't replace existing tools. It's just something new to help us see something in biology in a new way. And I want to point out that since a version of AlphaFold called AlphaFold Multimer was created, many different groups have been using this tool in the way that I'm describing, to do a screen, to screen thousands of protein pairs to test whether they interact. And those needles and haystacks of what they find, potential interacting proteins, leads to new advances in biology. So I just want to point out that this technology is being used by others in the world now to do these screens. And so that's why I became interested in also adopting this technique for my own lab's research here at the University of Utah. I'm not going to go in depth about what my lab studies, but I just want to give you a hint at what I'm using AlphaFold for my own research, we're really excited to find new hormones. These are molecules in our blood that coordinate systemic responses, like, for example, glucose uptake with insulin. And we'd like to use AlphaFold as a technique to simply discover new hormones and the proteins that they bind with, known as receptors. Because I'm very interested in understanding how different organs communicate in our body via these hormones. And actually, we've been screening, and we have this one protein we're really excited about studying in our lab. I'm not going to talk about it, but I at least wanted to show you a nice picture of it. We're calling it adiposin, and we found it in one of these protein-protein interaction screens, and we think it's a potential hormone. We also have a project in the lab that extends on this work to try to predict whether mutations in our genome in humans disrupts the interaction between this hormone and the receptor, and that would have really important implications for disease diagnosis, as well as the drugs that we give people that are trying to fix diseases that are due to imbalances in hormones, for example, such as diabetes. And for example, we're looking at this insulin receptor interaction, and we're systematically changing all of the amino acids on both insulin and the insulin receptor to see if we can have AlphaFold predict whether that breaks the interaction between the two. Okay, so I'm not going to talk any more about what my lab is doing, scientifically and biologically. Now I'll transition to telling you about my experience, running these screens on the Center for High Performance Computing, the CHPC at the University of Utah. So the main thing that I wanted to talk about was metrics of how long these screens take and why they take so long. So that's mainly what I wanted to talk about. So if the question is, how long does a screen take to do AlphaFold protein interaction screen, there are many variables that affect how long it takes. One is protein size. So proteins can come in many different sizes. They can be very small, just a few amino acids, or enormous, thousands of amino acids. And AlphaFold will take a lot longer to predict the 3D structure of a protein that's very large compared to a small one. These are some of our own, real-world timescales of how long it takes to predict a structure, depending on how big the protein is. Here's a 3,500 amino acid protein pair, and it took many hours. Whereas a very small protein pair, 600 amino acids, took only 7 minutes. But what's really important to stress is that the amount of time it takes AlphaFold to run proteins of different sizes is actually quadratic. It's not linear. The bigger the proteins get, it takes an extremely long time to run them. So the next variable is which GPU you're using. This is obvious. If you're using a new, fast, powerful GPU, it will run AlphaFold faster. But it was really important for us to try to rank all of the GPUs available here at University of Utah at the CHPC. I don't know if I'm going to explain it in this talk, but it is very helpful for us to know practically with our pipeline how fast our pipeline runs on the different GPUs, and then we've ranked them. So some of you may be familiar with the names of these GPUs here. The H100, which we have some of them here at the CHPC, that's the fastest to run AlphaFold. So that's the green bar here, and everything is compared to that. So you can see, for example, as you go down the line, it takes longer and longer and longer to run a protein pair interaction on AlphaFold. This is actually speed, so it gets slower and slower as you use GPUs that are older and less capable. And another thing that we've done is just simply cataloged all the GPUs that are available at the CHPC and where they are. This was really helpful for us in where we run our jobs. We need to know ahead of time which partition at the CHPC has which GPUs so that we know whether our jobs are appropriate to submit there. So just mapping this out was useful to us. But I'll say that what we do for these screens is we try to target GPUs that are owned by some lab on campus, so that when the GPUs are idle, then it's running our screen. And when the owners use the GPU, then our jobs are preempted, and they can be requeued and run again. And this is really useful for us because there's a lot of GPUs that are available here that are owned by someone that we can use when they're idle to do our screens. And it also alleviates a little bit of my guilt in using so many GPUs to do these screens. I try to avoid using the GPUs that are owned by the CHPC, for example. Another important aspect of how we run these screens is that, as I said, smaller proteins and larger proteins take different amount of times to run. So what we do is actually we break up our screens into little jobs. We parallelize our jobs, which is hopefully a familiar concept. But we make it so that some of our jobs only have small proteins, and we send those to less capable GPUs. Some of our jobs have large proteins that need to be processed, and we send those to the very capable GPUs like the H100s. And we also try to balance this in the sense that we send a lot of small proteins to the less capable GPUs and a very small number of large proteins to the more capable GPUs, so that hopefully our jobs all end around the same time. So yeah, if you're running a screen and you have to break that screen up into little parts, hopefully all the parts end at the same time, because otherwise you're waiting for a handful of jobs that are straggler jobs. And that really is when your screen ends, is when 100% of your jobs have finished. So managing all of these individual jobs to make sure that they're all running and that they all end as soon as possible is really the key of actually finishing these screens. So to give you an idea of how long these screens take, I just gave you some practical real world examples where we have 3 different screens we've run here. It shows the number of protein pairs we tested and how long it took. So here's one screen. We tested 20,000 pairs, and it took us 17 days. Here's another screen. We had the same amount of protein pairs, but it took us a month, showing you there's some variability in how long this takes. And our biggest screen has taken us 10 months to do, of 100,000 pairs. I think we could probably do this in 5 months at this point. This was a learning opportunity at that point. But something I want to stress, though, is that if you back of the envelope calculate based on these numbers, if say you wanted to do a screen of all pairs of human proteins, so humans have about 20,000 proteins, and if you wanted to do a pairwise interaction screen of all 20,000, it would take you millions of years with the current pipeline. And I really just am pointing this out because we can't just screen everything all at once with a snap of a finger. Like there are limitations in terms of the infrastructure that keep us from screening everything. And I'm not really complaining. I'm just pointing out that these screens take a long time. So finally, what I want to talk about is, I arrived here at the University of Utah 1 year ago, and I started to use all of the GPUs that are at the CHPC. And so I became interested in purchasing my own GPUs for my lab so that we could have GPUs that we could run around the clock and not have to worry about our jobs being preempted by other labs who actually own the GPUs. So what I did was I took a list of the GPUs, these 5 GPUs here that are available to purchase, that are recommended to purchase by members of the CHPC, and then I ran my pipeline on all 5 of these, which are already installed at the CHPC, to get a sense of the cost effectiveness. Like, yes, the H100 is faster with my pipeline, but is it more cost effective? And it's not. And so by ranking them in this way, basically the L40S became the most cost-effective GPU for me. The RTX 6000 and 5000 are more cost-effective than the L40S, but I was recommended to go with the L40S because it's more appropriate for a data center. Okay, so that's what we've purchased. It hasn't been installed yet, but it's a node with 8 L40S GPUs. And even though it doesn't run as fast as H100s, I get more bang for my buck. That's what I care about, especially in this funding climate. I think that's quite important to strategy to have. Okay, so I just have a couple more slides. I just want to summarize what I've said so far. My lab has a pipeline to do protein interaction screens using AlphaFold here at the CHPC. What we're doing is not different than other groups, but I really just needed to adapt doing these screens to the infrastructure here at the University of Utah. But hopefully, I think some of the basic tools, the bioinformatic tools that we developed, would actually be helpful for others at other institutions, particularly those using Slurm. Yeah, like I said, the protein size really is an important determinant for how we select the GPUs we're running on and how long it takes. We try to run our screens as many parallel jobs on a wide variety of GPUs that are mostly owned by somebody else, so that we are, trying not to take advantage too much of the resources. And that, this is not a complaint, but it's a fact that, we can't screen everything immediately. The current computing infrastructure limits us in the size and the number of our screens that we can perform. So we have to be careful in thinking about what we screen and how big, so that we're not too greedy and it doesn't take too long. So this is my final slide. Martin asked me to maybe comment on my experiences running AlphaFold at Harvard Medical School on their O2 cluster at their supercomputer. I used it for a short time before I came here to the University of Utah, and I don't have a lot of takeaways, but I think the main takeaway that I just wanted to point out, that something that they did compared to the CHPC that I really liked, was provide tools to monitor GPU usage of all of our jobs, as well as CPU usage. So it would be really helpful for me to be able to know for all of my jobs whether I am optimizing and maximizing the usage of a GPU or the CPU, so that I'm asking for the right amount of RAM for that node, or that the GPU that I'm using is actually appropriate for the job that I've given it. And I've tried to reconstruct some of these tools myself, but that's just a comment that I wanted to make, that I think it'd be great to have better CPU and GPU usage monitors at the CHPC. So that's it. I just want to thank my lab, many people at the CHPC who have helped me, including Martin and Drew, many different people here at the University of Utah. I do want to give a shout out to a previous lab member of mine, Arang Kim, who's at Harvard Medical School, who really turned me on to using these AlphaFold screens. And here's my lab website and my email if you want to contact me, take questions.
Great, thank you so much, Justin. And I put a link to your slide deck in the call document, so if folks want to reference that, that's there. And then please continue to use the chat to ask questions. And without further ado, I will pop over to Martin. Martin, did you want me to run your slide deck?
Yes, please.
Okay, I'm going to hit share on that.
Yeah, and so there are a few questions for Justin. So Justin, maybe take a look in the chat and see if we can answer those in the chat, or we can then. I would propose we have a discussion at the end, so that we can cover everything.
I'll start typing answers as well.
Thank you.
Yeah, what I'm gonna do is basically I'm gonna give the technical part of what's happening at or what Justin's using under the hood on our systems. I am here at Center for High Performance Computing at University of Utah. I've been here for a long time, and I kind of happened to discover AlphaFold probably 4 or 5 years ago. So Justin, you can go to the next slide. Because it looked interesting and we started getting requests for it. And so I set it up, and I was kind of appalled by the fact that it took 15 hours to do the first CPU intensive part, which is the MS8. It's multiple structure alignment search, which is basically searching these massive databases to find a match for the sequence that you're looking for. And then it took 30 minutes to run on a GPU to do the inference, which is sort of the magic piece that won AlphaFold and David Baker's lab the Nobel Prize, right? And so I kind of looked into why is that, and at that time we were storing these databases, which are a couple of terabytes, so they are not too small, on a relatively cheap JBot NFS file server. So that was one of the big reasons why it was slow. Then I tried the local disk, which we happened to only have spinners, and it was better, but it was 10 hours instead of 15 hours, so still pretty bad. And so, I kind of Googled around and found that this one tool that they're using for this database search called HH Blitz, they gave a hint to put the database indices into a RAM disk. And that really sped things up, and now the MSA search took like an hour, yeah? And so that kind of made me feeling kind of okay. So sure, we wasted an hour of GPU, but it wasn't 15 hours. So that was sort of our first release of the AlphaFold. Then I found probably 2-3 years ago a GitHub site where actually people did a very small modification of the AlphaFold code to separate the MSA part and inference part. Yeah, so you can now run two jobs, one on the CPUs only and the other on the GPUs. And so we created some aliases and basically provided users with the Slurm scripts to split these into two jobs. And so that's more or less what we still have. So, again, my hint here for how to make the best usage of the resources is to split the AlphaFold jobs into two pieces so you don't waste your GPUs during the MSA search. And then if you don't have a very fast file system, you do the RAM disk because the database indices are only about 20-30 gigs, so they are kind of manageable. Like these days the nodes have a few hundreds of gigs of RAM and it's not really using that much. So that's the AlphaFold, and still I was still not very happy at the time when we still were running everything on a GPU, even MSA search. So I ran into CaRCC, which is an alternative to AlphaFold with respect to MSA search. They're using a different program for that. And so I set that up as well, and we have a handful of users using that. By default, this CaRCC is developed by a research group in South Korea, so they're having their own server, which is serving this, and these MSA searches. And so by default, CaRCC actually does the MSA search on their server and then returns the result, and then the inference runs, the AlphaFold inference run on your GPU, right? And so I set that up using a local CaRCC, which is a command line version for the CaRCC. So we have that documented here as well. So by the way, these URLs kind of point to our documentation and discuss that. And then Justin came, and I go to the next slide. And Justin managed to get banned from the South Korean server pretty quickly. They do kind of put disclaimer, please don't overload our server because it's serving the whole world. So he asked us to set up the server locally, which kind of gave us a little bit of a head scratch, but because for good performance, they list that you need to have a terabyte of RAM and relatively fast disk drive to store these databases that they use for this MMSEX, which uses a little bit less size of databases. But in order to be performant, you need to have about 0.7 terabytes of the databases in RAM so that it runs fast. And so we did find these 10-year-old donated servers that were part of one of our oldest cluster. So I was like, well, people are running, I don't know, 30 gigabyte jobs on these 1 terabyte servers anyway. So we might as well use more of the RAM for something useful. And so we sort of did a prototype of this about probably 9 months ago now. And we finally are spinning up the real server right now for that. But really the trick here was to have the terabyte of RAM and then also copy the rest of the databases onto the SSD and symlink the RAM, the databases that are in the RAM to our NFS mounted VAST system, just because the whole database is like 1.7 terabytes. And so that's working pretty well, and it's actually very fast. I mean, the MSA searches on the CaRCC are probably order of magnitude faster than on the AlphaFold. They are a little bit less accurate from what I've read, but like few percent. So it's probably still acceptable to everybody, including Justin. Yeah? So that's more or less our CaRCC story. And with AlphaFold, they do have the MSA and inference split by default. So that's great. We're using that. The databases are not indexed, so we cannot use the RAM disk. And so we have everything on VAST, but these days we have VAST, which is a pretty performant file system. So we are actually doing pretty good on that. It's still a little slower in the RAM disk, the run the MSA on AlphaFold, but it's manageable. It's like hour and a half instead of an hour, something like that for this T1050. I think it's like 2,000 base pairs test protein that we have. The big change that necessitated us to make some changes with AlphaFold 3 is that they have more restricted licensing for the parameters, which is basically the neural network that is figuring out the structure. And so we opted for creating a separate group, for a Linux group for this, and we only allow users in the group after they get a license, they get access to to the parameters which we request through Google. Yeah, so we have that documented in here as well. And I just want to spend a couple more slides that there are other programs like this, right? So this is a really active field of research. We have recently installed this program called Bolts, which is also open source alternative to AlphaFold 3. The big deal with AlphaFold 3 is that it can process not just proteins, but other types of molecules as well. Yeah? So it's actually much more useful for all kinds of bioinformatics or biochemistry or whatever, any structural kind of studies that you do with bioorganic systems. And so Bolts is doing this too. It's open source, right? So it's kind of more attractive, and also it's using the MMSEX too, which is that faster MSA search like AlphaFold. They actually have an option to use the AlphaFold server. So we just had a first user that I'm dealing with right now of that. I installed it a few weeks ago. So just FYI, that exists. The Baker Lab, that's the other co-recipient of the Nobel Prize, along with the Google DeepMind people. They have a bunch of tools as well, which users are kind of requesting. So there's one called RF Diffusion, which we've installed. That one was a little trickier to install because it's set up for user space. So we had to, so basically, you pip install this to your home and you use it as yourself, and it's kind of hard codes path to your home. So we had to do some modifications to the code for the shared environment. Rosetta Fold was kind of a competitor to the AlphaFold 2 back in time. They have the all-atom version as well, so we're working on setting that up as well. And then finally, kind of the last slide, I believe. So we have a couple of researchers who actually enjoy doing their research in Google Colab. So there are notebooks to run AlphaFold, RF Diffusion, and Protein MPNNs, another tool from the Baker Lab, in a Colab. But they were not very happy with the 15-minute limit that they got on the T4s there on the TPUs that Google has on the Colab, and so they own a pretty beefy GPU node, and so they asked us to set it up in so they can run using Google Colab on their desktop, but running all this stuff on their server that is at our systems. And so it took me a little while, but I figured it out. So if you're interested in finding how to do that, let me know. We have a help page about it as well. I can sort of give you the soup on how to set it up.
Thank you, Martin. And I see lots of good questions for Justin and for maybe you, Martin, in there as well. So if you hit the chat, you can answer those questions. Our third speaker today was from Boston University, is Brian Gregor. So we have boxed you into only about 15 more minutes. Brian, did you make the call? There you are.
Yes, it did.
Yeah, would you like to share your screen, or would you like me to open up your presentation, however you'd like to do it?
Yeah, I can share.
Okay. And welcome. Thanks for making the call. You had a busy day, and Brian's jumping on. And again, folks, I was able to take PDFs of each of these presentations and I attached them so you can get at this content in the call document. So thank you.
Okay, so I just have a couple slides. There we go. Okay. So we had set up AlphaFold 2 a couple years ago.
Wait, Brian, who are you and where are you from?
Oh, sorry. Yeah. I'm Brian Gregor from Research Computing Services here at Boston University. Sorry, a little bit frazzled because I was on the 5th floor class over across the street. Had to make it here across 4 lanes of traffic, 2 subway tracks, and 2 bike lanes in like 6 minutes. So yeah, are you especially complex software like AlphaFold, we generally will do an install when people request it. So we've first installed a couple versions of AlphaFold 2 when that had first come out. And as sure all of you are familiar with, it came with a over 2 terabyte database, set of database files. Our approach, we stored those in a directory in our file system that is not backed up and is not part of our snapshot system. Just figured if anything ever happened, it's no problem to re-download it. At the time we were running CentOS 7, we used NVIDIA's Ubuntu Docker image, converted to a Singularity container, and then provided users with a wrapper script that would handle launching the container, setting various environment variables, setting the database paths, and so on. We use Sun, or variant of Grid Engine for our scheduler. So jobs, as they execute, have to stay within their requested resources. So one thing discovered had to do a bunch of customizing of the Python code so that it would recognize the fact that the user requested 8 cores and only run the database search section with 8 cores instead of attempting to use all the cores in the system, which would lead to their job getting killed. The first 2 installs we did with that, they had the exact same strategy. For the next 2, there's a paper done at PERC 2022, which is cited below, where one of the things they did was they took that database and put it into a Squash FS archive, which saves a lot of disk space. And they also found it actually improved performance because there's such a smaller quantity of data coming across the network when doing the database searches. Other than that, it was the identical sort of approach. When AlphaFold 3 came out, of course, the model parameters was the sort of biggest stumbling block. The fact that they can execute their main script in the CPU database phase, followed by the model inference, that's built in. It's a huge advantage. Our GPUs, of course, are in very, very high demand. So going and tying up a GPU node for 11 hours while you're doing a database search, followed by a drastically faster GPU phase, maybe 20 minutes or 10 minutes, being able to now run those on supercompute nodes as a supported feature in the software is very useful for deploying it. The database has drastically shrunk, only 600 gigabytes or so now. From the user's point of view, they have to provide customized JSON formats instead of just a FASTA file. I have not measured this, but my qualitative impression so far is that overall it executes a good amount better, faster than AlphaFold 2. So for the model parameter question, we took the approach where we simply push it on to the users to go and agree to all the license requirements and to go and download their own copy of the parameters for their own personal use. So every member of a research group would go through the same process and not share those parameters files. They're about 1 gigabyte in size, so it's not a storage issue. We do have a page that has some links into the various license files, some guidance on how to submit the form, so it goes through on the first try. We've not heard yet from anyone having issues using it. There's at least, we've had this installed since the very end of December. There's at least 10 people who seem to be routinely using it. I met with 2 brand new users yesterday and a research group last week, another like 8 users. So the interest is definitely there and seems to be growing. And everyone so far has gone through their processes and had any issues with it. If you go and ask for our module help on that the AlphaFold 3 module, it also will repeat the message that they need to understand the license terms and follow them. The installation here was generally much more straightforward than what it was for AlphaFold 2. One of the very first GitHub issues they had was actually a suggestion that, hey, why don't you use this Singularity def file and it builds a working Singularity container. So I just use that. The database, I still take the strategy of shoving it into the Squash FS file. Still saves a lot of space. The Squash FS file when the Singularity container executes, Singularity can automatically mount that into at a directory point inside the container. Singularity container itself is written with the Squash FS file. So again, the handling of that is easy from the container side. And again, it just saves a whole bunch of disk space. So we just continue with that. So it's about 200 gigabytes on disk now. Our wrapper script is more complicated. There's a bunch more parameters to play with. There's a splash attention parameter, gets set automatically based on your GPU. Again, in order to stay within our core limits, now we get this command line parameter to go and set the number of threads that these the jackhammer and endhammer processes use, except that those are hard coded to run with 8 threads. And there's a link, you can go see that in the code yourself. That command line parameter does not change that thread setting for those processes. So there's some handling in the wrapper script to go and figure out how to set that parameter appropriately, because it's more like it's setting the number of copies of those that you're going to run. So that was a little bit more complicated to handle that. The module, our provided documentation has been drastically expanded compared to the AlphaFold 2. We provide an example based on one of the example setups in their GitHub repo that sets up the 2-phase calculation as different jobs and show the users how to go and make the GPU portion wait for the other job to finish. The input JSON file format is biggest change from, I think, the user point of view. They do document it online. I haven't come across a lot of examples or guidance online for this. I did have one ticket come in where someone had a valid JSON data structure, but it was an invalid JSON data structure for AlphaFold, so they were quite confused by that. Finally got it figured out. Considering how complex this input file can be, you can have a gigantic, like FASTA file sequence embedded in there. DeepMind doesn't provide any kind of validation tool except running AlphaFold on it, which seems kind of a bit of an oversight. There's no, like with XML files, you can have like a defined schema and you can validate your file format against that to make sure you conform. DeepMind hasn't given us anything similar that. And from experience with that user, if you have a bad input file, the error messages are don't really tell you anything particularly useful. So I expect to hear more from users in their input files as usage and interest continues to grow around that particular topic. But so far, the rest of our documentation examples seem to be working for. That was a lot of fun.
Great. Thank you, Brian. So we have roughly 10 minutes left on the call, and there's been a lot of content thrown at us over the last 50 minutes. So thanks to our speakers, to Justin, Brian, and Martin. I don't know, Justin, did you see any thing you wanted to elaborate on after Martin's talk with all the different questions that came through?
I think the thing on my mind is how I adapt what I've done for AlphaFold 3. So previously, the screen I told you about was AlphaFold Multimer, which is based on AlphaFold 2. And I've only played with AlphaFold 3 briefly, thanks to Martin, and found that it was quite slow and unclear how I would adapt my parallelizing strategy to AlphaFold 3. So that's what's on my mind. And actually, Brian, you said that AlphaFold 3 ran similar or faster than AlphaFold 2. Was that running AlphaFold 2 using CollabFold or just the regular?
Just on our cluster, the database phase certainly seems to be generally faster, but there's also a huge reduction in the database files, which I suspect is due to that. And the on the GPU side also seem to be faster. Again, I haven't specifically done a head-to-head comparison and measured it. The 2 users I met with yesterday have been experimenting with bolts, and for the particular work that they're doing, they're using some novel ligands that won't be in the AlphaFold databases. They found so far that running bolts is overall they'll get to an answer faster than they will at AlphaFold 3. But
yeah, let me add to that. I believe that's because Justin, you're using CollabFold, so it's using the MM6 for the MSA instead of the Jackhammer HH Blitz. Yeah. And so the MM6 is much faster. especially if you have all the databases in the RAM. Yeah, so that's what we have on our server, and that's what they have on their server in South Korea as well. Yeah, so we don't really have 2 terabyte server anywhere which would be able to put all the AlphaFold databases in the RAM. And I did get, as I said, get pretty good performance boost by using those database indices in the RAM disk. That ate about 30 gigabytes of RAM, but it was still much slower than the MM6 through the CaRCC. So the bolts actually might be a good opportunity for you, Justin, to do the all Adam kind of simulation, because it's using the CaRCC, the MM6 as well, the CaRCC.
Great. That gives me a nice path for my next steps. I'll try bolts.
Great. There was a few more questions in there. Martin, did you see the questions from Jim and others in the chat there? Has anybody tried setting up a generic JSON blob people can use to customize?
Yeah, pretty much. I just show users the sample JSON that Google came up with for testing, and then they are on their own. We really haven't had any issues, but I don't think we had a big uptake on AlphaFold 3. I think we have like maybe 8 users that we allow the access to the tool so far.